But nobody prior to the paper that we published last year had speculated that basal metabolism is changing, and that might be a driver of obesity.
Hi, this is Tucker Goodrich, and I have the honor today to be joined by Professor John Speakman, who is a professor at the Chinese Academy of Sciences in Shenzhen, China, and at the University of Aberdeen in Scotland. He is a world-renowned expert on the topic of energy balance, with a particular interest in what's known as doubly labeled water. And I believe it's correct to say you run the world doubly labeled water database, which is the topic of the paper that we're going to be discussing today in part.
Yeah, right. So there's not many people use this technique. And a few years ago, they came together and decided that we would pool all the data that we had into a big database to kind of answer questions that individual studies couldn't afford to look at. And that was taken on by the International Atomic Energy Agency and is a sort of publicly available resource that people can write in and say, I'd like to do this analysis. And then the data that's necessary to do that's released, as long as nobody previously proposed the same analysis.
Right. So how did you get interested in this topic? I mean, it's not the sort of thing that, say, an 8-year-old boy would say, I want to grow up and study energy balance.
Yeah,
I presume.
right.
Maybe you did.
No, I didn't either. So yeah, so my background is in zoology. I did a zoology degree for my undergraduate, and as it came to the end of that program, we did a project, and I did a project with a guy called David Bryant, and he was interested in using concepts of energy balance to understand adaptation. So why animals do what they do, how they survive winter, how they breed in the summer, and things like that. And I did a project with him about wading birds, food intake in wading birds, and just loved it. Just really liked it, and sort of saw that it was a powerful way of understanding what's going on more than just kind of watching animals in the wild and things like that. So I ended up doing a PhD with him, also on wading birds. So my background was completely in animal science. And then when I got my first postdoc, I continued using the methodologies that I'd learned during my PhD in another project on bats. And then I eventually got a faculty position. But the thing that became pretty obvious was that it's quite difficult to get money to work on those things. So although they're super interesting, they're also extremely difficult to raise funds for. So I started looking around at other things that energy balance is important in, and the two things I kind of dropped on were aging research and obesity research. So I put in grant applications to do projects on those things, and both of them were successful. So I kind of had a kind of didn't really have a dilemma what to do, because I had to do both because I had the money. And so my career sort of flowed along those different tracks. But the thing that and I still do, some ecophysiology work in collaboration with people. But the thing that really cements them all together is this kind of interest in energy balance and how energy balance is important for understanding all these issues.
Right. So that's energy balance for the audience. I mean, the simplest explanation of it is the phrase we've all heard, calories in, calories out, which is true as far as it goes. Ultimately, that's what it boils down to. But the interesting part is that it's not just that simple. There's all sorts of regulatory factors going on in the body, and there are, different, everything you eat doesn't equate to a calorie. Some things are used for structure. Some things, can be, I guess structure is probably the major place that things would go to in a non-energy context. So, I mean, it's fair to say that a lot of that is not really well understood. There's a lot of conjecture about how much, I mean, one term I keep coming across in my reading is the idea of feed efficiency, where, this is mainly in agriculture, where you're trying to figure out what is the cheapest feed that you can feed to an animal to get it to gain weight the fastest, because that makes the whole process the most profitable. So there's a lot more going on than a simple calories in, calories out formula.
So
Is that fair to
I
say?
think, yeah, I mean, I don't disagree with that. I mean, I think the problem is that the way it's often stated makes it difficult to accept that argument. So when people say calories in, calories out, that's total. The problem is, as a straightforward physical statement of what's going on, it has to be true. And so basically it's difficult to then say, yeah, you're right, that is a load of. So the thing is, the whole regulatory system that controls how many calories we consume and what happens to those calories is a very, very complex system that we don't fully understand. And to just sort of try and say, well, okay, I'm going to summarize that whole system with this little phrase and then say it's rubbish is really not kind of the way to do things, I don't think. So I think
Well,
it's a
it doesn't add
distraction.
anything. It doesn't, coming from an engineering background, neither part of that really adds any value to figure out what's going wrong.
Right, right.
Yeah, which is what, for all of us, is really the interesting part of this question right now. I mean, academic interest aside, in the middle of a global obesity epidemic, why is that happening? Figuring that out is of paramount importance, I think, and I presume you feel the
Yeah,
same way.
absolutely. So, I mean, from my perspective, I mean, I've heard people say, well, it's too late now. There's 45% people are living with obesity, and 75% of people are living with overweight and obesity. So it's too late. There's no point in knowing how it happened. What we need to know is how to reverse it. But the way I look at it is a little different, because my perspective's a little different because I'm living in China. And if you look at the map of obesity in the world, obesity is actually still really just a problem of the US, Europe, and the Middle East. And there are enormous parts of the globe that have still not really been touched by this epidemic. So if you look at the whole of Africa and the
Right.
whole of Asia, they're still at the very start of it. And so if we understand what went wrong in the US and Europe, then we stand a chance perhaps of doing something about it exploding in these other countries. But it
And
started to
hopefully,
happen already.
hopefully, if we figure out what is causing it, then maybe we can reverse it where it's already happened. I mean, I think it's
Yeah, I mean that's possible, but of course
right.
the route to reversal doesn't have to be the same as the route to causing it in the first place. I mean, it would be
True.
nice to do that, but I mean, if you look at the latest drugs that seem to be making a dent on obesity, like the GLP-1 receptor agonist, the cause of obesity was not because of too much GLP. So it's the route out of
Right,
it
right, 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 right.
is a different route than what got us into it in the first place.
Right, right. Yeah, no, that's a fair point. There could be a useful distinction between treatment and causation.
Right.
Yeah, I agree with that, because I mean I've talked, we both are part of an online community, obviously, where there are lots of people with lots of different opinions. And one gentleman I talked to treats obesity, and I asked him how often his approach doesn't work. And he said, well, it always works, but some people won't do it. And for those people, and I mean, fair enough. I mean, there are some people who, we know what causes lung cancer, and some people keep smoking. So,
Right.
a treatment for lung cancer would be a useful thing. A treatment for obesity would be a useful thing, because
Yeah.
it's going to
So I mean,
benefit some number of
pretty
people who
clear.
just, you know, won't do it any other way.
If you have a treatment, it's not going to cure anything if you don't take it. So I mean, I think
Exactly.
that's a pretty clear situation that we're in, and adherence is an issue with most sort of dietary intervention type approaches. That people don't adhere to the diets, and that appears to be pretty much independent of the approach. So it's not like that some diets are really easy to adhere to and other ones are not. It seems pretty much that all the diets have the same kind of pattern of adherence.
Right. Well, yeah, which gets into other questions of human nature. I mean, as as someone who had a problem with overweight and now adheres strictly to a diet for reasons other than overweight, you know, it keeps working so long as I keep
It
doing
keeps
it.
working as long as you stay on it, and
Yeah,
that's the same
exactly.
with the
And
drugs, and I think this kind of thing with the drugs that, people are sort of saying like, oh, wow, when you stop taking the drug, the weight comes back. What did you expect?
right. Well, that's
There
like
is
an
no drug
alcoholic
in
complaining,
existence.
well, can I stop drinking for a little while and then start drinking again? No.
Yeah.
You'll be right back
So,
where you
so
started
there's
off.
no drug in existence that keeps
Right.
working after you stop taking it. And in fact, that's a good thing, because we don't, let's imagine you're one of the people who gets adverse responses to this drug. You don't want the drug to last forever. You want to be able to stop it under your own control because then that'll stop the side effects.
That's a
So it's
phenomenal point.
it's it's not a bad thing that it stops working when you stop taking it.
Okay, so bringing up this topic of all these different diets brings us to an excellent starting point to the ultimate conversation, the most interesting conversation that I would like to have with you today. And this was a 2018 paper that you wrote called Dietary Fat, but Not Protein or Carbohydrate, Regulates Energy Intake and Causes Adiposity in Mice.
Yeah.
And in it, you basically looked at three different models of obesity, if I can say that. You looked at something called the protein leverage hypothesis, which is very interesting. You looked at the classic idea that fat is what makes you fat, which is the reason why we all adopted a low-fat diet, or at least part of the reason. And then you also looked at the idea that carbohydrates cause obesity, which is, low carb diet as a treatment for obesity has been very popular. That doctor that I just mentioned who said he has great success with it, that's his approach. And so describe what you found in looking at these three different dietary approaches.
So what we did was the first step in this project was actually to design the diets because there are a lot of mouse diets out there, but they're not particularly well controlled. And so the first sort of step of the study was to say, okay, what we're going to do is we're going to change the proportion of fat in the diet, which was the thing that we were particularly interested in. But previous mouse diets, when they change the amount of fat, they also change the composition of the fat. And so that leads to lots of kind of confounds. And in some of the situations, you had like the fat was increasing, but also some components of the carbohydrates were increasing as well, like maltodextrin. Yes. So then you couldn't separate out whether the fat, if the change in the weight was due to the fat or due to the maltodextrin. So we had to go in and we worked with Research Diets, the company that make rodent diets, and design these diets so that the composition of the fat was constant and equivalent to the standard American diet in terms of its composition of omega-3, omega-6 fatty acids, and the proportions of saturated, monounsaturated, and polyunsaturated fats. So we did that, and then we changed the fat content. But then we had a total of 29 different diets in which we manipulated the fat content and also the carbohydrate content and also the protein content. So by doing that, allowed us to kind of have this matrix of diets that we could then test these different ideas. So
And
the first,
I just want to interject here. What you described sounds, I hate to say it, pretty rudimentary, but it's not. It's practically revolutionary in the area of
so
rodent
so the point
diets,
is, people
having gone
hadn't
through
done
many,
that
many,
before.
many of them, and now having a database of all the different diets that are used in all of these different studies. Often you can't compare, not only can't you compare one diet and one study to another diet and another study in any easy way, but you may not even be able to compare it to the control diet, which may be just chow
Right.
unhelpfully.
So we wrote a paper recently in like Metabolism pointing out these issues with the diets that very often the control diet is completely uncontrolled. And so
Right.
there's no idea what the animals are actually feeding on in the controlled condition, which seems crazy, but that's a very, very common aspect of these nutritional manipulations. So while it seems like an obvious thing that you would think this was already all done, actually it's not, and very few people had that kind of level of control. So one of the people who has also done this with a big matrix of diets is a guy called Steve Simpson in Australia, and
Hmm.
he actually was the originator of this protein leverage hypothesis.
Right.
So the protein leverage hypothesis, many of your listeners are probably familiar with it, but just in case they're not, the idea there is that we don't eat food for energy, we eat food for protein. And so the situation is that if you decrease the percent protein in the diet, then you have to eat more of it in order to get the same amount of protein. And so what that basically is saying is that the protein is leveraging the total energy intake. So let's imagine you go from 30% protein in the diet to 15%. To get the same amount of protein, you have to eat twice as much. And that means you overconsume calories, and that drives positive energy balance, which then leads to adiposity. So what we did in that study was have diets that change from 30 down to 5% protein at two different levels of fat in the diet. And in both cases, at the two levels of fat, we found absolutely no effect. So there was no change across that range in the amount of body fat deposited, the total food intake, the energy expenditure. The animals were clearly eating the food for its energy content rather than for its protein content. And so we
Were they
concluded,
now if they were getting as low as 5%
yes.
protein? Were they actually getting into protein deficiency at that level, or is that can a
No,
mouse survive at
I don't think
5%?
so. They seemed fine. There were no sort of big
Okay.
behavioral problems with them. And since then, actually, we've done another paper that was also published in Cell Metabolism, where we took the protein levels down to 1%. And actually, in those animals, they lose weight rather than gain weight. And that loss is actually a mix of lean tissue and fat tissue. But also, even there, even at 1% for 12 weeks in a mouse's life, is not enough to cause any sort of deficit. So they have better memory performance, they're better on a rotor rod machine, which is a sort of measure of agility. So it seems not. I mean, five
Okay.
percent was fine for them, but there was no indication of any protein leverage going on in terms of body fat mass.
Okay, interesting. And then you looked at carbohydrate.
Yeah. So
Um,
then the carbohydrates model and the fat are kind of inverse of each other. So the idea is that uh, if increased carbohydrates are making or making mice put on more body weight, then you would expect this relationship to the carbohydrate content, but not to the fat content. In fact, you would expect higher fat, which is then balanced by lower carbohydrate, would make you thinner. And we studied this over a range of fat and carbohydrate content. So the fat contents range from about 8% up to about 60%. And over that range, there was a positive relationship. But if you looked at the relationship to the carbs, there was a negative relationship. So as you increase the carbohydrate content of the diet, then the mice were thinner. If you increase the fat content, then the mice were fat.
and was that a linear relationship with the increasing fat content?
It wasn't. So what happened was it increased up to about 40%. So there was an increase in body fat mass up to about 40% fat in the diet. And then it was stable between about 40 and 60%. And then at the very highest fat that we used, it was slightly down, although in the analysis that wasn't significant.
Right. But you did not in this paper get into ketogenic levels of fat consumption.
No, nowhere near. So what's interesting is we got a lot of kickback, obviously, from people who were saying, "Well, that's rubbish."
When you're goring everybody's oxes, yes, you would expect some kickback.
So what we've done since that paper in work that's not yet published is we've extended the fat content. So we've gone up to levels of about 95% fat in the diet, and we've taken the carbohydrates right down to 1%. And in fact, in the very latest one, effectively 0%. So what happens then is quite interesting, because that sort of flat section between 40 and 60, and that slight decrease as you go above 60, that slight decrease continues down. So what happens then is, if you're eating a diet that's effectively a ketogenic diet, although it's hard in a mouse to force them into ketogenesis,
which is an interesting topic on its own, but let's just leave it at that.
Yeah. So it seems that mice are pretty good at creating carbohydrates from protein, so they can switch up gluconeogenesis, and so
create
they
their
don't
own glucose,
need
right?
to create. So they don't need to create ketones in the same way that we do. So they do increase the ketones, but it's not as profound an effect as it is in humans. But the point is that what we said in the paper was dietary fat makes us fat. It's not carbohydrates and it's not protein. And I think for the data we had at the time, that was correct. But clearly since then, it's not a linear relationship, and it doesn't continue above that sort of flat zone between 40 and 60. And in fact, once you get up to these very high levels of fat and very low levels of carbohydrate, there's no real drive towards adiposity. So
right, which as I recall, you had shown in an older paper that a ketogenic diet, for some reason, seems to have some unique property where it's, unique in a high fat diet property where it's no longer adipogenic and no longer promoting obesity.
Right. So I mean, I think that previous paper you're referring to was less controlled than what we've done now. So what we've now done is kept the composition of the diet more constant, and again keeping exactly the same fat composition as we change the relative proportions of things in the diet, and that's sustained. So the interesting question then becomes, why do we have that pattern, and what are its consequences? So I think,
and before we go into that, I would just like to observe that this is the change that we've seen in lots of human populations over the 20th century and into the 21st century, where they go from, particularly in places like Asia or Africa, they go from a very high carbohydrate diet into what we've called the nutrition transition as they industrialize, and their fat consumption goes up along with. So this is not just a mouse study. You're replicating what we see in human populations. I mean, it's just a mouse study, but it also happily replicates what we see in human studies.
Yeah, right. So I mean, I think if it was, if we didn't expect some correspondence between what's happening in mice and what's happening in humans, there would be no point doing it. So there is that kind of underlying kind of thought that, maybe this does have some relevance. But of course, it is just mice, and we need to be a bit cautious about what's going on there. So what we did in that original WHO study was we looked what was going on in the brains of the mice in terms of the sort of signaling network in the hypothalamus. So the hypothalamus is the thing that controls our food intake. And what we showed is that as the fat content of the diet increased, then they were switching on all these kind of reward systems. So the endogenous opioid system, the endogenous cannabinoid system, serotonin signaling, all these things are kind of going up. And so basically what it seems is that the animals just like the diet. So it's like they have a sort of reward when they feed on that diet. And actually, it's quite interesting. If you look at the time course of what they do when they're exposed to the diet, as soon as if you take a mouse that's eating 10% fat, and then you put it on a diet with 50% fat, what happens is the day after it's put on that diet, it enormously increases its food intake. And it sustains that for about 3 or 4 days. But then it slowly declines over time. So it's kind of like it gets this reward hit. And then it's like anthropomorphizing the animal kind of realizes if it continues doing that, it's going to be in trouble. And there's some kind of feedback that kicks in and it starts to reduce its food intake.
Well, but that's also similar to drug consumption, where you have an initial strong effect of a drug, and then over time, the body, I mean, opioids, this is what happens, part of the food reward
Yeah,
system.
okay. So you have to take more to get the same kind of hit idea.
Right.
But in this case, they don't like further increase their consumption. What they do is they actually tone down their consumption. So they're actually eating less and less over time. And one thing that's really, really difficult to understand is the amount that they reduce. It would only need to be slightly more and they would avoid the obesity problem. But for some reason, they can't just eat that little bit less. They still eat just a bit too much, and that's enough for them to keep accumulating body weight. And that's always perplexed me. Why can they not just eat a little bit less of what they do?
Well, you and every human dietitian alive.
Yeah. I mean, it's happened. It's a small. So if you think about it, what happens is it shoots up for like 3 or 4 days, and then it starts to come down. So after 15 days, they're eating substantially less than they eat at 5 days.
Right.
So the question is, why at 5 days can they not reduce to the level they're at 15? And at 15, they're eating much more than they are at 30 days. So why at 15 can they not eat less? Because if they did that, if they just dropped down to the level that they eat at 30 days, they would have no problem.
But then when you take them up into the ketogenic levels, what happens then? Does their actual gram consumption drop down?
So yeah, so basically what happens is initially they just eat the same weight. So they eat the same weight, which means they get a bit of a calorie surge, and then they reduce what they eat. So they then come back into balance, and then there is a small effect, but it's not significant when you put it through all the stats. So it seems that they kind of instantly recognize what the issue is, and they don't overconsume. So it seems that there's like a rewarding section, this kind of golden zone where you've got 40 to 60% fat in the diet. And if you can think of it like a mountain, and people climb up that mountain as they become obese. So
right.
the question then is, how do you get off the top of that mountain? What do you do to reverse your body weight? And some people go down the keto route, so they increase the amount of fat and they lose weight because they're going off the mountain that way. But other ones reduce the amount of fat and they come off in the same direction that they've gone up. So the interesting thing is that there seems anecdotally to be some kind of difference in those responses. So it seems that if you go off in the keto direction, you don't seem to get as hungry as you do if you go off in the high carb direction. So that's anecdotal. I don't know whether it really pans out, but it is a kind of interesting observation that maybe there's something different about coming off that mountain in the two directions.
Right. Yeah. My daughter, in the United States, we have this phenomenon called the freshman 15 or freshman 40, depending on how unfortunate you are, where you go away to college and you put on 15 or 40 pounds or whatever. And my daughter always had a little bit of a weight problem when she was in the United States. And then she went to college and discovered the freshman 15, except she went to Japan. And in Japan, the freshman 15 is the 15 pounds that all the foreigners lose when they arrive in that country. So I start getting these panicked emails from my daughter saying, I don't have any clothes to wear. They're all too big.
Well, that's a nice problem to have.
Well, it's a better problem to have anyway. It still means a new wardrobe, but anyway. But I mean, it was fascinating. And I said, what are you eating? And she said, I'm eating more rice than I've ever eaten in my life. I know the Japanese, there are lots of differences between the Japanese and the American diet, and that is certainly one of them, but it was fascinating what a stark difference that was. And, 6 years later, she's leaner than she ever was when she was here in the United States.
And on a high carb background, but a
Yeah.
low fat. So
Yeah.
so I mean that's like a classic situation that she's coming off the top of the hill, and she's gone down it in the high carb direction. But I think the problem here is this kind of polarization that people have come off it in the sort of keto direction because what they've done is reduce the carbs in the diet. The obvious thing to say is, well, it's the carbs that makes you fat. But if you
Right.
come off that hill in the other direction, then it's the fat that makes you fat. And actually, the reality is it's neither. It's the combination in that particular ratio.
Maybe.
I think.
Okay. So then we get into, okay, let's put a marker on that one and come back to that because that's a very interesting point, I think. So then we get into your more, by the way, I want to commend you. I don't know if it was your idea or Cell Metabolism, but at the beginning of this paper, you have this wonderful graphical abstract, which I've used countless
Drawn in
times.
Amsterdam Airport.
Beg your pardon?
Drawn in Amsterdam Airport.
Really? By you?
Yeah, I had a delayed flight and I had a couple of hours and I spent the 2 hours doing that graphical abstract.
It's brilliant. I mean,
Yeah, thanks very much.
every single, I mean, probably not every paper would lend itself to this, but it is absolutely brilliant that you put this in your paper. And I think I've already will have used it in this discussion at this point, so everybody will know where it comes from. So then back to the energy balance side of your career and the doubly labeled water. So just to summarize this quickly, doubly labeled water is a way of using isotopes in water. You feed them to people, and then you can use the excretion of those isotopes to calculate how much energy they're using. And this
that's right.
method has been around for several decades at this point, right?
Yeah. So actually, it was originally devised by a guy called Nathan Lifson in the 1950s. And
so many decades, okay.
Yeah, but the problem was that getting the isotopes in the 1950s was pretty difficult. So he had to, like the very first paper that was done, he had to go to Los Alamos and beg them to give him some isotopes to work on, and he only had enough to study one mouse. So that was the kind of state of the field in the early 50s.
But one mouse.
Yeah, absolutely. So for between 1949 and 1955, there was only one animal ever studied. But then, and right up to the 1970s, it was still relatively small numbers of animals because it was expensive to buy the isotopes.
Right.
The very first human study was done by a guy called Dale Scholler, who was at University of Chicago at the time, it's now in Wisconsin. And he did the first study in humans in 1982. But then it was still pretty pricey. And so we only started to get reasonable numbers in the late 80s. So around about 87, 88.
Okay. And you're using this to better determine how energy and food sources are being used in the body. And I mean, I guess there's a spectrum. At the most accurate end would be the metabolic chamber, where you're measuring everything that goes in and everything that comes out, poop and pee and exhalations, and measuring everything that goes in. Very expensive. I mean, Kevin Hall, who you've written a number of papers with, does that at the National Institutes of Health, and even they can't afford to do it for more than a couple of weeks because it's so expensive to do.
Yeah,
so that's
right.
The extreme.
So the problem is it's not only expensive, but it's also gives an answer to a different question. So the question that we're kind of trying to address with the doubly labeled water method is how much does it cost to live your daily life? So the things
Right.
that you do in your life are not things that can always be constrained inside a small chamber. So,
Right,
going to a
because with
club,
doubly labeled water, you give people the water and then they go out. They're not in a
right?
metabolic chamber and they go out and do
Yeah,
whatever it is they're going to do. And you don't have to feed them or bother them or do anything, just measure the excretion periodically.
yeah. So
And
what we
then
do
and
is,
then just to give people the understanding where this sits in the spectrum, then at the other end of the spectrum, you have food frequency questionnaires and measuring body weight and all this stuff, which is grossly inaccurate and prone to all sorts of memory recall problems. So this is like, I mean,
but
it's
not only memory recall problems, there are like structural issues within those approaches. So if you think about 24-hour dietary recall, for example, I might recall accurately that I ate an apple yesterday. But what happens when that's converted to energy is there has to be an assumption about how big that apple was. So they then multiply that by a standard 70 kilocalories. But, I may have eaten an apple that's 120 kilocalories.
right.
That's not coded in the thing. So we can't blame the inaccuracy all on people's memory deficits. There are other
Well,
structural issues with those approaches.
yeah, absolutely. I mean, just and you wind up with a situation, my understanding is, correct me if I'm wrong, especially in obesity research, where fairly small changes over each day can lead to obesity, and those can be dwarfed by measurement errors when you're talking about food frequency questionnaires.
Yeah, I mean, I think food frequency is not routinely being used to estimate energy intake. So it's more concerned with uh, just trying to understand what the pattern of dietary intake
Right.
is. Whereas 24-hour recall is used to try and estimate the total energy intake. So that's trying to do the same as what these other techniques like indirect calorimetry in chambers and double-labeled water is trying to do, but measuring the other side of the equation. And I think the experience there is that these tend to be pretty inaccurate tools and uh, prone to bias in relationship to body weight of the person who's reporting. So what happens is that there's more underreporting in people that are bigger, and consequently, for a long time it was thought there was no difference in intake between people who have an obesity issue and people who don't. But that's clearly nonsense because their energy expenditure is considerably higher. So they must be at least eating enough to cover their energy expenditure. So it became clear, principally because of this doubly labeled water technique, that couldn't be true. So through the 1990s, that was generally the idea of the reason why people were becoming obese was not a food intake problem, it was an expenditure problem.
Right, right, right. Okay. So doing all of this work led to you creating this database that we mentioned at the beginning of this discussion, and th that led you to write this fascinating paper, Total Daily Energy Expenditure Has Declined Over the Past 3 decades Due to Declining Basal Expenditure, Not Reduced Activity Expenditure. And you came up with this observation by looking at the data in the doubly labeled water
Right,
database,
right.
correct?
Yeah.
Describe how that came about.
Yeah. So, I mean, of course, there's been a lot of interest in. If you ask anybody what causes the obesity epidemic, generally they have two answers. One is it's fast food, so they're thinking about the energy intake side of the equation, or they say, well, it's because there's been increasing sedentariness in our lives, and we're not as physically active as we used to be. And that reduction in our energy expenditure has created this positive energy balance. So if you were going to study that, the
The old
best way to,
back to the for another simplistic phrase, eat less, move more, right? It must be
yeah,
because
well,
we're not
but
moving
that's,
more.
yeah, so that's,
Right.
And I think they're not completely unreasonable ideas.
Well,
So I mean,
it's calories in, calories out
yes, it
restated. It
is.
has to be
It's
true
it's
ultimately,
using
but
energy
it's
balance to understand why there's a problem.
right.
Okay, so I can't argue with the logic of the idea. Okay,
It's
but
just not explanatory.
well, I mean, it could
It
be,
doesn't tell you why it's happening. It tells you the
yeah, right.
mathematical relationship that
But let's imagine that there has been a reduction in our physical activity, and there is some evidence to support that. So if you look, there was a paper by Tim Church, for example, which looked at work time activity and showed that there'd been, since the 1960s, there's been the shift in more active workplace activity to more sedentary. So there's more people working in offices, there's more people doing more sedentary jobs. And he estimated that that change was probably about half a megajoule. So it's like about 140, 150 kilocalories a day.
right.
So it's possible, I mean, and there's
Well, and it's
certainly,
yes, and everybody sees that in their daily life. So we always, I
yeah,
mean, my grandfather was born on a dairy farm and died a professor. So he alone went
so,
that from one
yeah,
end to
right.
the
So
other.
so I mean, it's plausible, and there is observations also that uh, people who are living with obesity are less active, so that's pretty much known. So there is a kind of smoking gun there, but it all has to work through energy expenditure. So it's not like there's some magic link between your activity and your body weight. It's got to be acting through this energy balance equation. And if we'd been around when the doubly-labeled water technique had just kicked off, if at that point we'd said, I know what would be a really good idea because there's going to be an obesity epidemic. Why don't we measure a group of people at random from a town somewhere, and we just measure them every year, and we measure a different sample every year? We can see what the decline in energy expenditure is because nobody had the prescience to do that.
Right.
But the second best way to do that, which was done in 2008, was there's a guy called Klaas Westerter who works in University of Maastricht in the Netherlands. And he'd done lots of studies back to the 1980s. So I contacted him and said, look, you got all these studies, all of them have a control group. Why don't we just pull out all the control groups for all the studies that you've done? And we can quantify this reduction over time in the energy expenditure. So he said, yeah, okay. So I went over to his lab and we extracted all the data. And there was no change. So we looked at that between like 1985 and 2005, and there was nothing, absolutely flat. But that wasn't kind of ideal because in Maastricht, the obesity rates only gone from about 5% to 10% during that same interval. So,
You may just be
maybe
missing the signal.
yeah, right. So it wasn't an ideal data set, and it wasn't an enormous number of people, it's only about 300 people. So then when the database came along, we had a lot more data, and we could look at the problem in a lot more detail. So we had about, I think in the end, about 2,000 data points. And what we could do is we could look back to the late 80s up to about 2017, 2018, and we could see whether there was a signal there when there's a much bigger data set, but also over a time period when the rate of obesity are much higher. So we restricted it to US and Europe, and then we looked at what had happened, and there's a signal there. So there's a reduction in our energy expenditure over that time period. So that's great. But then, the assumption would be if we'd only had that data, the assumption would be, well, that's it. It must be our declining physical activity that's caused that reduction in our energy expenditure. But actually, we could test that because for a lot of the subjects, we also had the basal energy expenditure measured in a metabolic chamber. So if you subtract the basal metabolism from the total metabolism, what you're left with is generally ascribed to be the activity metabolism. So it's not exact, and there are some complexities in that calculation, but give or take, it's a measure of physical activity, energy expenditure. So what we did was we plotted that against time, and actually that's gone up slightly.
Oops.
So that was very unexpected. But of course, if that's gone up slightly and the total's gone down slightly, then what that means is the basal has to have gone down. And when we plot that, that's exactly what's happened.
And now, this is again, for the audience, basal metabolic expenditure isn't something that ought to be changing. I mean, that should be in some large part genetically determined, right? I mean, it's the difference between a warm-blooded animal and a cold-blooded animal is the level of energy expenditure.
Well, the difference between, okay, some humans might be a bit cold-blooded, but I think
fair enough, but let's not get into personality.
That difference across the sort of phylogenetic spectrum is not the differences we're observing between people.
Okay, fair enough.
You're correct that you would not expect. There is enormous variability once you take the weighted factor, but there is a lot of residual variance that we're not really clear where that's coming from. But nobody prior to the paper that we published last year had speculated that basal metabolism is changing, and that might be a driver of obesity. Right. So this was like a completely novel suggestion, and it wasn't, I mean, it would be nice to say, okay, we had this great idea that maybe basal metabolism had changed. We didn't have that. We just were looking for the signal about activity. That signal wasn't there, but what fell out of the analysis is this decline in basal metabolic rate, which was completely unexpected.
Right. Now, there was, just kind of an aside here, there was a paper looking at the Chimene, a indigenous population in the Bolivian Amazonian jungle, where they noted that their body temperature has been declining over time. And that's, it's a very healthy population. They've been kind of the poster child for, how a traditional diet results in very low levels of cardiovascular disease. So they've been fairly well studied, I think it's safe to say. Could this be related to the effect that we're seeing in those populations?
So what's interesting is if you look, there's data on body temperature in the US going back to the Civil War, and actually in the US, body temperature has also declined over time. And what we did was, because we got this result with the basal metabolic rate, it only went back to like the late 1980s. And so the question was, well, does that extend further back in time? So what we did was a literature review going back to the early studies of Benedict and Atwater in the early 1900s. And that signals there across that whole period. And it corresponds to the signal in the body temperature. So it seems like these two things are related to each other. As you drop your basal metabolism, you're producing less heat, your body temperature is dropping slightly over the same period. So I think they're sort of intimately related to each other.
And of course, that's intimately related to the increase in body weight, which can be, if you go back and you look at Civil War soldier recruits, they had a lower BMI on average than modern men do. And it's been not a flat linear increase over time, but it has been a pretty steady one.
Yeah, so I don't know if you saw, there was a recent paper by Torkild Sorensen who looked at these populations in Denmark where they have really, really nice data because they're all kind of conscripted into the army. So for males, they have fantastic data through time. And basically, he shows that the antecedents of the obesity epidemic are much earlier than what people generally consider. So the start of the epidemic is the 1910s, 1920s, not the 1950s, 1960s.
Right. Although it does seem to have kicked up. Obviously, there's a big dog leg.
And it's more obvious, obviously.
Yeah.
So, I mean, it's more obvious, but it's still there. The signal's there early on. So it's something that changed early on. It's not the advent of fast food in the 1950s, that's not the thing that kicked this off. It was something earlier than that.
Right. So then putting these two papers together, the obvious thing to look at for what's causing these effects is the fat consumption, because the clear signal here is obesity. It's easy to see. And we note that there's been, and you note in your paper, the Blasbalg paper from the NIH that looks at the change in fat consumption over the 20th century in the United States. And that's happened around the world due to, the uptake of what I like to call an industrial diet. And describe that for us, because I have my own thoughts on this, but this podcast is an interview of you. So
sure. Okay. So here's my take on it. So, I mean, it's obvious there's been lots of changes in the diet, and one thing that we were particularly interested in is the composition of the fat, but it's not necessarily that's driving this. But so it's only a hypothesis at the moment. But it is very clear.
It's important to note again that in your mouse paper, you used varying amounts of fat, but a constant composition of fat across those varying amounts.
So that's you looked at strength, but also a
snapshot of fat across what humans have done over the last 100 years or so.
Right. So what's happened in humans is if you look back to the early 1900s, then probably 90% of the intake was butter and lard. And the total fat intake has increased through time. But at the same time that the total's going up, the amount of butter and lard is declining in absolute terms. But in relative terms, is declining even greater, obviously, because what we've loaded onto the diet are things like soybean oil in particular, but also other seed-derived oils have increased enormously. So we have this dual thing going on, that the total's going up, the absolute amount of fat from animal sources is going down. And then the difference between those two is this big expansion in plant-based fat.
Right. And it's important to note that calories have also gone up over this period.
Yeah, absolutely. If the total fat's going up, the calorie intake's going up. So one thing that we were interested in is what the effects of composition of the diet might be on the metabolic rate. So the problem with this is that it's pretty difficult to do it in humans. So we did a mouse study, and this was a collaboration with some guys from Yale. And what they did was they exposed mice to a matrix of diets, again, that varied in their fat content and measured their metabolic rate. So what was interesting was that the only fat, when they looked at total intake of saturated fat, total intake of monounsaturates, and total intake of polyunsaturates, the only factor that came out as important was the saturated fat. And the saturated fat intake was positively related to the metabolic rate. So the more saturated fat you had, the higher the metabolic rate in the mice. So one potential idea then is that the decline in the saturated fat intake through time since 1910 has been a factor that's caused the decrease in the basal metabolic rate over the same interval. And that can be a contributor to the obesity problem over that same kind of range of time. So that's the kind of idea that we're working on at the moment.
Right. But again, this is an association.
It's in mice. Well, I mean, in the mice, it's a causal relationship because we're manipulating the diet, that we're looking at the energy expenditure that comes out of that.
Okay, fair enough. So right, and this is how we do it. We find an association in humans, and then we go to mice to try and figure out what on earth is going on here, because people are too annoying to study.
So in the humans, it's correlation. There's obviously lots of other things happening. So the interesting thing is that there have been a couple of studies that have attempted to manipulate the diet in humans and then look at what the consequences are for the metabolism. But they tend to paint a much more variable and confusing picture. So there was a study actually involving that guy, Klaus Westerter, that I just mentioned, where they fed individuals polyunsaturated fat. And what I would have predicted is if you bump up the polyunsaturated fat, you would get a decrease in metabolic rate, but actually they found an increase.
Now,
so then,
okay, go ahead. Sorry.
So there's a couple of other studies. There's another study that showed no effect whatsoever of manipulating the fatty acid content of the diet. And then there was an interesting study from the 1990s, which showed it was not the composition of the fat, but just the total fat. And that total fat, if you increase the total fat in the diet, there was a reduction in the metabolism. And that would also be consistent with the data that we have going back to 1910. So the total fat consumption has gone up. Maybe it's the total intake that's driven down the metabolic rate rather than the specific changes in saturated or unsaturated fat.
Interesting. Yeah, so it's not as clean a picture as we would hope for. What?
Well, it's just a picture, it's there's no kind of hope at the start of it. It's just looking for these relationships, and seeing what drops out.
Did the group at Yale come up with any mechanism by which, I believe in your paper you said it was palmitic acid specifically, how that could increase?
Done some work. Yeah, so we'd previously done some work. So there is this idea. Actually, one of your previous guests was instrumental in deriving this idea. So Tony Holbert from Australia came up with this idea, the membrane pacemaker hypothesis. So his idea was that if you have like the phospholipid membrane on a cell, if you have lots of polyunsaturated fats in there, then the membrane becomes leaky. It can't hold out protons. And so what happens is you get some sort of leak across that membrane, and that affects the metabolic rate. So the idea behind that would be that you eat a certain diet, that diet becomes reflected in your membrane composition. And because of the leakiness of the membrane, you get a change in the metabolic rate. But actually, the prediction from that idea is the opposite of what we observed in mice. So you would expect the more PUFAs you have in the membrane, you would actually have a higher metabolic rate. But actually, what we observe is the more palmitate in particular you have in the membranes, the higher the metabolic rate. So it's opposite the membrane pacemaker hypothesis.
Right, because part of that was getting to longevity and the idea that basically the hotter the furnace burns, the shorter your lifespan is going to be. And there's some pretty good evidence for that across different species of animals, although there's, again, a lot of variation.
So I did a lot of work on this about 20 years ago. And in fact, the associations between lifespan and metabolic rate all hinge around covariance with body weight. So if you remove the body weight effect, actually that signal disappears. So there's very little evidence for a relationship between metabolic rate and lifespan once you take body mass out of the equation.
Okay, so larger animals just tend to live longer.
Yeah, exactly. So they live longer and they also have lower metabolic rate. And so it's just, bigger animals have lower metabolic rate. There's an association between metabolic rate and longevity just because of that size effect. So it's kind of like, I wrote a paper once with this in it that, they also have bigger eyeballs. Bigger animals have bigger eyeballs.
Okay.
And so there's a relationship between eyeball diameter and longevity.
Sort of like the old sharks and ice cream, right? Shark bites are correlated with ice cream intake, but
right.
There's no mechanism there.
Yeah, so what I said was, and if you really believe this, you can dream up a mechanism that, animals with bigger eyeballs can see predators coming, and that's why they live longer, because they can run away. And they also have longer legs, enabling them to run away better.
Right. Okay. Fair enough. Now, one point that Tony brought up in our discussion and in his book, he has a book out, Omega Balance. I don't know if you've seen
No,
it
no, I
yet.
didn't know. Right.
Okay. It's very interesting. He definitely, you both, amongst perhaps many other things, you have in common that you were both zoologists who have become fascinated with humanity.
Yeah.
And he's kind of going the same pathway. And in his book, one, he goes so far as to say that he wouldn't even use PUFA as a demarcation of types of fats, that he would say it comes down to saturated fats, monounsaturated fats, and omega-3 versus omega-6. And a number of the papers, so just to quote one interesting sentence to me from your paper, as linoleic acid is desaturated to form arachidonic acid, and arachidonic acid is linked to endocannabinoids, it has been speculated that expanding linoleic acid in the diet may be linked to various metabolic issues. However, effects on BMR are disputed, and if anything, PUFAs lead to elevated, not reduced metabolism, 35, 36, although many studies suggest no effect, 37, 38. What you don't get into so much in your paper is the distinction between omega-6 and omega-3. Some of the
Sure.
papers that you refer to get into that distinction, and I was wondering if you have any thoughts on that.
Yeah, so I mean, main omega-3, I guess, like monounsaturated fatty acids, and so there's been a lot of consideration of things like oleic acid on various aspects of weight regulation and diet, and they generally come out suggesting not much of an effect. So we didn't
Not much
particularly
of a
look at.
pro-obesity effect.
Yeah, exactly.
Right, which is, given that it's one of your body's favorite fats after palmitic acid, isn't super surprising, to me at least.
Yeah, so I mean, it just in the mouse study that the Yale guys did, it just didn't come out. Yeah,
Right.
so this omega-3, omega-6 difference didn't seem to be a major driver. The main thing that seemed to be driving it was the just the amount of palmitate. That seemed to be the key.
Okay. So did they specifically look at omega-3 versus omega-6, or because one
So they
paper,
looked.
sorry,
So initially, they broke it down by all the fatty acids that were in the diet initially, and then they grouped them into saturated mono and polyunsaturated fats after that. But they initially looked at all the individual amino acids, fatty acids, and there was no signal in there apart from palmitate.
okay, okay. Because I don't think you got into that level of detail in your paper.
No,
Um,
it's not in there, but that was in the analysis. So the
okay,
problem is that once you start to analyze it by individual fatty acids, the power is much reduced because you've got the same sample size, but you get many more predictors.
right,
Yeah. So if you then group it as to three predictors, you have much more power.
right. The old, the tyranny of the p-value.
Yeah. I mean, it's just if you have 10 predictors,
Right,
you know,
yeah, right. It's just,
it's
it comes down to arithmetic
numbers.
at that point, right?
Yeah. So
The
you
more
need to
data
do
you
many,
have, the
many
bigger
more
this, yeah,
animals, yeah, which
right.
which they didn't have. So it didn't make sense to include that analysis, but it doesn't there's no massive signal there.
Okay. Okay. Well, that's very interesting. Okay. So what future work do you think is required to determine the validity of this saturated versus, I mean, looking at the human diet, the saturated versus polyunsaturated signal and the effect it's had on energy balance, and metabolic rate to start off with, and then ultimately obesity, which is of course the question that looms large over all of us at this point.
Yeah. So at the moment, we have a couple of studies planned. So we have a big mouse study where we're going to use 11 diets that range from, so we've designed the diets so that monounsaturates are constant across all the diets. And then the fat content is fixed at 40%. But as we go from one end to the other end, we're dropping the saturates and replacing them with linoleic acid.
Right.
So
the
then,
main fat in seed oils.
Right. So we have then the main outcome from that I'm interested in is the energy expenditure. So as you drop that saturated fat, is that really driving a change, an increase in the basal metabolic rate? But also there's this idea that because of the sort of link between linoleic acid through to arachidonic acid and anandamide.
The endocannabinoid system and food reward.
Yeah, basically, it's potentially the case that they could be stimulating the food intake. So we're measuring both aspects of the change in the energy balance, the change in the energy expenditure, which we think is probably driven by the saturated fat, and potentially a change in food intake, which is driven by this conversion of the linoleic acid into things that bind into the cannabinoid receptor. So the idea is to do that experiment, see what we get, and then if we get a big increase in the food intake in relationship to the linoleic acid, the next step is to knock out the cannabinoid receptor and see whether that persists. So basically, if that increase in the food intake disappears, then we have the mechanism by which linoleic acid might be driving greater food intake. Right. But if we knock out the receptor and that persists, that suggests that linoleic acid is having an effect independent of the mechanism that a lot of people think it might be having. But I should emphasize, we have no evidence yet at all that this change in linoleic acid has changed the food intake, because we didn't do the experiment yet. So it may be there's no change. So we'll just have to wait and see what the data looks like. So that's why we didn't schedule to do the cannabinoid receptor mouse until we know what's happening with the food intake in the first place.
Right. And you've done a number of, not recently maybe, but a bit of work with rimonabant, which was a drug that was a endocannabinoid receptor antagonist, if I'm getting this correctly, meaning it blocks the effect of the endocannabinoid chemicals on the receptor. And that's got some effects on, that's got some effects on both obesity. It was at one point a human anti-obesity drug until it got pulled from the market. And then it's also shown to have some effects on energy expenditure.
Yeah, small, but it's there. But I think the interesting thing was why rimonabant and these other ones were pulled, because they cause people to have suicidal thoughts. But nobody actually committed suicide. It was a psychological effect. Whereas if you look at bariatric surgery, people die, but it's still allowed. So I think there's a difference in the playing field.
Yes, we replaced a drug where people would think of suicide with a surgery where people actually commit it at much higher levels than anybody ever saw from rimonabant.
Right. So nobody died from rimonabant, as far as I understand. But people do die from surgery and complications afterwards or whatever. I'm not sure.
And from committing suicide after they have bariatric
really? Okay.
surgery.
I wasn't aware whether that increases or not.
Oh yeah, Eric Topol, who was doing a cardiovascular examination of rimonabant when it was banned and had to stop his study, wrote a commentary on that I think he said that the suicide rate from bariatric surgery was like, an order of magnitude higher than what was even proposed for rimonabant.
Yeah.
So yeah, we're definitely on the same page there. That was a mistake of medicine writ large. Managing the whole system of medicine, they, focused too much on one particular drug and not on what all the alternatives were. But okay, so you've done some research on that. I mean, that's going to be an interesting paper. Any idea? You hate hearing this, but any idea when it'll be out the door?
Well, okay. So if you think about the experiment, the main experiment is going to take about 6 months. So that's scheduled to start in late February. So we will know probably the outcome of that first experiment by October.
Cool.
So then if there's no effect on the food intake, there's no point in doing the cannabinoid receptor knockout mouse. So then we'll just aim to publish that straight away.
Right.
If there is an effect, then we'll have to do the cannabinoid receptor knockout mouse. So actually, we've already scheduled to build the mouse. So we will have mice available and be able to breed up those colonies reasonably quickly. And we'll, obviously have an indication what's happening with the data before the end of the experiment. So we'll know whether to really go for that. So assuming that we can start that experiment round about the end of this year, it will be summer 2025 before we have the result of that. So then it would be written up and submitted. So we're looking fall of 2025 before that's out.
Right.
It's slow.
Yeah, so lovely.
So the other thing that we're doing is actually this is only a plan at the moment. That's a grant application, but what we're planning to do is a couple of things. So first one is a cross-sectional study where we bring people in, just measure the fatty acid composition of membranes in the red blood cells.
Mm-hmm.
So that's an indication of what their diet is without, falling into all these problems of dietary recall and stuff like that. And then measure their metabolic rate in the chamber. And so we would then have a correlation, hopefully, between what's happening in the membranes and what their energy expenditure is. And then the second phase of that is to do an intervention study where we manipulate the diets of the people for a period of about 3 weeks. So the reason to do it for 3 weeks is that's about the lifespan of a red blood cell. So if you change the diet for 3 weeks, you should get a signal in the red blood cell population. And then look at the change in metabolism in relationship to the change in the fatty acid composition.
Do you think that's going to be, I mean, that's long enough to change the red blood cells without question. Do you think that's going to be long enough to change the metabolic rate throughout the organism?
If the sort of idea that we're working on is that it's the change in the membranes that are some way affecting the metabolic rate, and that could be through this leakiness idea that Tony came up with, but also, one idea is that the rigidity of the membranes affects the proteins that are embedded in the membrane and affects their ability to function. So I mean, it's possible that it could have those effects in a relatively short time frame. So if you look, for example, at the study that I mentioned previously, they did a manipulation for 14 days and got an effect on the metabolism. So I think we're probably safe.
Okay, because I mean, you're looking essentially at mitochondrial membrane composition at that point.
Yeah, right. Which may be difficult to nudge, but well,
I mean that I've seen lots of papers where that responds, in rodent models again fairly quickly to diet, but it tends to vary across tissue. So that's why I ask if you think you'd see an organism-wide effect.
Yeah, so I don't know. So we previously did work in mice where we looked at membrane lipids, and they're pretty highly correlated across tissues. And so if we pick a signal up in red blood cells, it's probably going to be indicative that there's been a change across the board.
Yeah, no, that's a fair assumption, I think. So now you've
So paradoxically, that would be quicker to do than the rodent experiment. So we'll have the human data. If we get the grant, we'll have the human data before we have the mouse data. So that'll be an interesting situation.
well, that'll make the people who complain about mice data very happy, I suppose.
Yeah, but my only worry is that we'll have the human data, and then when we submit the mouse paper, somebody's going to say, "Oh, this has already all been done in humans. Why do we need a mouse paper?" We need a mouse paper because we just spent 2 years doing the experiment.
Well, and also you're gonna get a lot more, you're gonna get a lot cleaner data out of a mouse experiment
Yeah,
than you
there'll
are
be
out of
more
the human
mechanism as
experiment.
well. So, yeah.
Yeah. Where Okay. I could ask a zillion questions about that, but I'm not going to go into it because I would be just nerding out and indulging myself. So I'll spare you that and
Sure.
my audience. Um, that's fantastic. So now, one other thing I would like to touch on. So you are an expert in energy balance and thereby in obesity. I mean, I know energy balance is your main focus, but you recently organized an obesity seminar in London and you had a bunch of
Yeah.
beg your pardon?
Yes, I'm agreeing.
Yeah, yeah,
I
yeah.
did.
And you had a bunch, clearly they all came there because they think they see you as a leader in this field and they were interested to
Well,
participate in your
remember there were
thing.
4 organizers.
Well,
So
still. Okay.
David Allison, Torkel Sorensen, Kevin Hall, and myself. So it certainly wasn't me that was the draw.
Fair enough. Fair enough. Deuterium and chronic disease. And you're also an expert in the effects of deuterium on, or using deuterium to measure these processes. Now,
Sure.
there's a hypothesis going around that deuterium is what is driving, let's just say the simple deuterium is driving chronic disease. And that, I've looked into this a little bit and, you know, A, like a lot of these things, there's a natural level of deuterium in the environment, just like there's, a historic level of fat in our diet. And it seems that, if you get too little deuterium or too much deuterium away from the natural level, that you start seeing positive and negative effects on organisms. So it's an interesting hypothesis. Have you seen, but you've been spending quite a lot of time pumping substantial amounts of deuterium into these organisms, including people. Do do you think that there's anything to this hypothesis? And if not, why not? And if so, why?
Well, okay. So I mean, I mean, I saw all the stuff, and obviously we're using deuterium, like you said, and we're putting quite a bit of deuterium into people. So I was really interested in whether that was having negative effects, and also, uh, what influence that kind of hypothesized physiology might be having on the technique. Is that affecting how accurate the technique is? Does that explain some of the individual variability and the validation studies of the technique? So I did look into this in some detail, and my conclusion is that it's to be honest, it's not. It just doesn't hang together in any way as a story. So I can sort of
And yeah, why? Can you elaborate a little bit on why?
yeah. So I mean, if you look at the document that was produced by that guy Laszlo Boris, who's the main guy who's driving this. Uh, what he says is, so he's talking about the dangers of changes in natural abundance of deuterium in compounds. And so what he says is that if you take fatty acid, for example, that has got these levels of deuterium in it, then that can create a problem because when the fatty acid is oxidized, and those hydrogens appear in water. And what he says is that if you have a deuterated water molecule, that somehow destroys metabolic nanomotor in the mitochondria. So I don't think there's any evidence whatsoever that happens. But let's just imagine that that's true. That if you have a deuterated water molecule, then that has some negative effect in the mitochondria.
So what he's saying essentially is, just for the audience here, he's saying that complex 5, which is basically a little rotary. Yeah, I mean, it looks like a little rotary engine, and it produces the ATP that we all understand is the energy currency of the cell. And he's claiming, and clearly breaking that is a bad thing. So he's got that. But
right. So I think the first thing is there's no evidence that deuterated water molecules break that motor. So I think that's I couldn't find anything anyway. So that's a kind of an assumption that's been made. But let's give him that. Let's assume that's correct. Okay. So what you have to just think about is the quantification of these effects. So basically, the background level of deuterium is 155 ppm.
Parts per million.
Very, very low. Okay. And he says that some fatty acids may be enriched by a factor of 4 in the levels of deuterium. So in other words, instead of 150...
Is there any evidence that happens?
I couldn't find anything for that either.
Okay.
But there was some evidence in an amino acid, so in a really unusual situation. So some people looked at deuteration of amino acids in collagen extracted from seals. And what they found was, for some reason, and I think it is pretty interesting biology, I have no idea how it happens, but for some reason, proline had 4 times the background abundance of deuterium.
Okay, which tells us that maybe seals concentrate deuterium and proline for some reason we don't yet understand.
That's
fine,
that's right.
Feasible.
Which is interesting. Interesting. But what he then says is, well, let's imagine that these fatty acids that are being eaten in modern society are also deuterated to the same extent. So they're 4 times higher than the background level. And I don't think there's a shred of evidence to support that, but let's also go with that and see what that means. So if you just work through the numbers, you can imagine what this is. So if you think about a background level of 155 ppm, what that means is if you take a fatty acid like stearic acid, then what that means is for every 170 molecules you have, one of them in one location will carry a deuterium molecule. Okay? Whereas if it's 4 times higher, that means 4 out of 170 will have a deuterium molecule. So the vast majority of molecules are not carrying a deuterium. And even within those molecules, the majority of hydrogens are not deuterium.
Right.
So then wind it forwards and say, okay, what happens if we oxidize one of those fatty acids? If we imagine that actually the fatty acid is at the regular background level of 155, then when it's oxidized, what that means is that 1 out of 6,000 water molecules that come from that oxidation process carries a deuterium. Okay? Whereas if it's 4 times higher, it's 1 out of 1,200. But if you think about it in terms of percentages, what we're saying is that if you oxidize a fatty acid at natural abundance, then 99.98% of the water that comes from that is undeuteric.
Well, and the
Was
implication of this would, if his hypothesis is correct, is that the body would have to be incredibly sensitive to small increases in the amount of
yeah.
deuterium in
So
the body.
the increase is just unbelievably small. So you're going from 99.98% being unduterated to 99.94% being
Right.
unduterated. So that
So,
change, even if we give him that, there's a fourfold higher level in the fatty acids, which there's no evidence for, and the idea that a deuterated water molecule kills the nanomotor, which there's no evidence for, even if you accept those things, the magnitude of the difference is so small, it's just completely not going to be of any physiological importance.
but if it was, and if he's right, then a bunch of scientists who are running around pumping organisms full of deuterium over the course of a number of decades might notice something.
Yeah, right. So I
So,
mean, I'm obviously a shill for big deuterium here because I don't want there to be any effect of deuterium going on. But I mean, I think realistically it's not happening, and there's a test that you can make for it. So I mean, he claims that all these changes actually in the deuterium level are actually having a negative effect on cancer rates and stuff like that. So the interesting thing is that what happens with the natural abundance level of deuterium is it's not fixed. So the reason that it's not fixed is that when you evaporate water, the activation energy to evaporate water is greater for deuterium than it is for water with just two protons on it.
so
So the
evaporation
consequence.
is a distillation process then.
Yeah, right. So what it does is it enriches the amount of deuterated water. So because if you look across the planet, and this has been known since the 1970s, that if you look across the planet, the natural abundance of deuterium is much higher in the tropics than it is at the poles.
And that's
So what
because
would
water tends to evaporate from the tropics and then flow north and precipitate
right,
out with lower deuterium levels.
right. So that global cycle and the amount of evaporation that's happening in the tropics means that people in the tropics have much higher natural abundance than people living at higher latitudes in both directions. So if its idea is correct, that you get a protection because of low deuterium levels, then what you would predict is rates of cancer would go down with increasing latitude.
And obesity and heart disease and all the
Yes,
other chronic diseases,
and they don't.
they go up.
So obesity, there's no difference. But cancer, so I looked at a couple of papers on links between latitude and cancer rates just to kind of check it out. And prostate cancer in North America increases with higher latitudes, and breast cancer in Australia increases as you go further south. So in both those cases, it's the opposite direction. Obviously, for lots of other reasons that influence cancer rates, but the deuterium effect is not a dominant signal if it's there at all. So I come to the conclusion that this is really not something serious, and we shouldn't be particularly worried about uh, taking doubly labeled water because I've done it myself a couple of times.
Right. And, I mean, yeah, when I first heard this, my immediate reaction was, okay, what's the process by which this is getting so enriched? And I have, as you said, I also haven't been able to find any explanation for how it's happening or a quantification that it is
But
happening. I did find some interesting papers. I mean, there is definitely an area in cancer research looking at deuterium depleted water because it stimulates oxidative stress on the cells, and there's a whole range of different therapies that are used against cancer that increase oxidative stress. But the flip side of that is they've looked into deuterated lipids, specifically like linoleic acid, the PUFAs that are subject, and DHA, the PUFAs that are subject to oxidative damage, and thereby induce oxidative stress. And what they find is that the deuterated PUFAs induce less oxidative stress because they're less chemically active, which is,
Right.
there's a lot of evidence, which we won't go into here, that oxidative stress is a factor in all these chronic diseases. And if this deuterium hypothesis is correct, then a simple test of deuterating polyunsaturates and feeding it to organisms should produce bad outcomes. But what it seems to do, and there's not a lot of data, but what it seems to do is reduce the bad outcomes. So the exact, again, reverse of the hypothesis that they're
again,
proposing.
you just got to think about this numerically as well as just kind of practically. So when you give a deuterated, an artificially produced deuterated molecule, all of the hydrogens are deuterium. So we're not talking about one molecule in 170 stearate molecules. We're talking every single carbon in every single molecule is
You mean
deuterium.
every single hydrogen?
Every single hydrogen,
Right.
sorry, in every molecule of stearate is a deuterium. And that's why you get some physiological effects at those levels. But that's, like it's like literally several 100,000 times more deuterium than the signal that we see in the environment.
Right. And a similar amount more than his proposal of a fourfold enrichment.
Yeah, right. So this is like a 400,000 fold enrichment.
Right. And it's beneficial. I mean,
right. Okay. So it goes in the wrong direction anyway.
Yeah.
But I mean, it's not, you can't compare those things. So he sort of said, if you look at the phase transition between deuterated and undeuterated molecules, there's a big physiological effect. Well, of course there is, but you never ever see molecules in nature that are completely deuterated. These are completely artificial compounds.
Right. Okay, well, that's good. So we can stop worrying about that one. If we had, obviously, both of us.
I was glad he brought it up because it's like something to worry about when you use these techniques, but it's good that it turns out it's probably not important.
Well, especially if it's bollocksing up the results of your experiments. I'm sure you were not. I mean, that's why you would take it seriously. Oh my goodness.
Yeah, absolutely.
Yeah. Okay. Any anything else you'd like to touch on, or?
No, I'm good. I think we had a really nice discussion. So thanks very much for inviting me to come and speak. And it's really great to be able to kind of air our ideas to a wider audience. And I hope people follow our work and understand what we're trying to do. And that's great.
Great. Yes. Thank you so much for taking the time to do this. I really appreciate it. It's kind of funny, when I try and do research in this area, when we started communicating over Twitter and email, and I started trying to do research in this area, you've written a lot of papers. It's hard to pursue some of these avenues without coming up with John Speakman again and again and again. So I really appreciate you
I have
taking
a big group though, so it's not, I have a lot of people in the group that generate a lot of work. So it's great. I really enjoy what I do, and if it makes a contribution, then fantastic.
Yeah, no, it's a really interesting and I think essential avenue of research that you're pursuing. So thanks for taking the time. And
Thanks very much for inviting me. That was fun.
Great. Terrific. Thank you very much.